I would like to thank Ron and Kathy for inviting me to talk today. I know I am holding you up from this poster session and all of the hors d'oeuvres, so I'm going to thank you as well for sticking with it on the very last talk of the session. So I was actually quite pleased because a number of the speakers before me really touched on several of the topics I'm going to talk about today. So I think I can breeze right through why we're studying statin pharmacogenomics. Karen told you that it is one of the most widely prescribed drugs for the treatment of cardiovascular disease. We just heard about it. I just wanted to spend a minute to talk about how this drug actually works because I'm going to talk about some mechanism later on, so I want to make sure we're all on the same page. But statins are competitive inhibitors of HMG-CoA reductase, which encodes the rate-limiting step in the cholesterol synthesis pathway. That reduction in intracellular cholesterol upregulates a receptor called the low-density lipoprotein receptor, which is the major mechanism by which plasma cholesterol can then move from the periphery into the liver and therefore, of course, reduce the potential for it to build up in the artery wall. And so we know that statins are very effective in terms of reducing plasma cholesterol. This is data from the cholesterol and pharmacogenomics clinical trial that Ron Krauss ran a number of years ago, in which almost 1,000 participants, 2/3 Caucasian, 1/3 African American, were treated with simvastatin for 6 weeks. And you can see that while on average there was a 41% reduction in plasma cholesterol, there's quite a wide range. There are some individuals for whom the statins had no effect, and there are some who had really dramatic reductions in cholesterol. And so the clinical implications of this variation in response, I think, is best illustrated by this idea of the number needed to treat. And the number needed to treat is an epidemiological measure which calculates the average number needed to prevent one bad outcome due to the treatment. And so here I'm just showing you for illustrative purposes data from the West of Scotland coronary prevention trial in which about 6,500 men were treated with pravastatin for 5 years, and this is primary prevention. And here they found that after 5 years, that among 42 statin users, one of them you could attribute did not have a heart attack due to the drug. 5.5 still had a heart attack despite the fact that they took the drug. The vast majority did not have a heart attack, but it was really irrespective of whether or not they took the drug. And then when you overlay on top of that the idea that we know that there are adverse effects, as Karen mentioned, 11% may develop diabetes, 10% may develop myopathy, and really the concordance of the risk for myopathy and diabetes as well as efficacy, we don't yet understand how those risk factors may be related to one another. And so the coloring I have up here is really for illustrative purposes only. But clearly, when you look at this, you want to be this one guy here in the green for whom the statins were effective and not the person for whom the statins were ineffective. You had the heart attack and then you had some sort of adverse effect. And so there's really a clear motivation for us to try to identify markers of statin response. Because Karen really talked about work to look at adverse effects, today I'm going to tell you more about the efficacy side, which, as you heard, is a little bit more challenging than the adverse effects, but I think we're making some progress. So one of the tools that we use to try to identify genetic determinants of drug response is a panel of lymphoblastoid cell lines. And so from that cholesterol and pharmacogenomics clinical trial, we had generated about 1,000, these are Epstein-Barr transformed lymphoblastoid cell lines, and we've devised a protocol in which we can expose them in vitro to an activated simvastatin or sham buffer for 24 hours, and then we can measure all sorts of cellular phenotypes. And one of the nice features about this is that you can consider each of these cell lines as sort of little mini models of the donor individual, right? Now at this point, we have made the environment exactly identical, or we've tried to make it as identical as possible across all these cell lines. And so variation that we're seeing in the cells really should be a reflection of genetic variation of the donors. And so we can measure things like gene expression and intracellular cholesterol and expression of the cell surface LDLR protein. So all things which are really relevant to the mechanism of action of statins. And then we can actually test for correlation between variation in our cellular drug response and variation in how well that donor performed in that statin clinical trial, because we have measures like plasma cholesterol, plasma triglycerides, glucose, etc. And we have been very successful in using this type of strategy to identify genes and pathways that we believe to be involved in statin efficacy. So I sort of put the punchline in the title of my slide, in that today I'm really going to tell you about two stories which, when we started them, we didn't think that we would converge on long non-coding RNA, but as you'll see, that's where we ended up. And so just to give you a very brief survey, in I think one of the most comprehensive analyses of the human transcriptome that was reported last year, this was an analysis of over 7,200 RNA-seq libraries that were really ranged across a wide swath of tissues and cells from normal and diseased individuals, using not just aligning all of those sequences to the known transcriptome. They can assemble all of these sequences to actually identify that there are 91,000 expressed genes, of which the vast majority, as you can see in the red here, are long non-coding RNAs. So 68% of these are long non-coding RNAs. And whether something is called a long non-coding RNA is really just defined by the fact that it's not thought to be coding, and it's larger than 200 nucleotides. So that's about as working of a definition as one could have. You can see that there are some other types of transcripts on here. So let's see. TUCP, these are transcripts of unknown coding potential. So these are situations in which there's been some algorithm that suggests that these might be protein coding, but in fact there's no physical evidence. Pseudogenes I would really call a type of long non-coding RNA. They just have a lot of homology to the original gene from which they were derived. And read-through transcripts are special situations in which you have a transcript that actually reads through two different gene units. And so depending on whether or not that open reading frame is maintained, these guys could be coding, but they're probably likely not going to be protein coding. So actually it turns out that really only 23% of the human transcriptome encodes for protein coding genes. And I think one thing that was really striking to me is that of the ones that they identified, the vast majority of them, 79% of these guys had not been previously annotated. And so clearly, when we're thinking about this issue of how do you know the gene is a gene in that GWAS locus, this becomes a really important thing to consider. And from this analysis, they found that 7% of the long non-coding RNAs that they identified overlapped with disease-associated GWAS SNPs. So this transitions into the very first story, which is how we were trying to really annotate a GWAS locus and stumbled upon a long non-coding RNA. So as I mentioned, we're interested in statins, and a number of years ago, the Jupiter clinical trial, this was a trial of about 3,500 rosuvastatin users who were treated. 3,500 rosuvastatin users. This was a placebo-controlled trial, and a GWAS analysis was performed, and there were a handful of genome-wide significant hits, but these were all really well-defined loci. Most people would not argue about the gene annotations, but they also reported this table of subgenome-wide hits, and I've highlighted in yellow the one that I was particularly interested in on chromosome 6, and you can see, even though it's subgenome-wide, it has still a pretty defendable p-value of 10 to the minus 7, which is pretty good in pharmacogenomics. And the reason why we were interested in this is because one of the gene annotations was this gene called IDLE, or MyLip. And IDLE stands for inducible degrader of LDLR. And as I told you at the very beginning, one of the major mechanisms by which statins actually lower plasma cholesterol is by stimulating upregulation of the of the low-density lipoprotein receptor. And so we thought, this is like the best case biological candidate that you could identify. And IDLE had not been previously implicated in statin response. And so you can just see right in this picture that IDLE is involved in ubiquitinating LDLR, and so it reduces LDLR protein levels. Okay, so where's the SNP? Here's the SNP that was identified in the GWAS locus, and it's about 10 kb downstream of MyLip IDLE. And if you look at this is LD in the Caucasians, it actually is within a very, very tight LD block. And surprise, surprise, it sits right on top of a long non-coding RNA called RP1 13D10.2, which I will just call RP1 because that's an awful, awful gene name. I haven't yet figured out who gets to decide when you can rename a gene, but I think that when you've identified some sort of function, I think you should have the right to rename a gene. Okay, so the first question we asked was, is there really any evidence to link this GWAS SNP with RP1 or IDLE? And so you want to know, is it an EQTL? And so at the time that we were looking at this, there wasn't really a lot of good data for where we could see RP1 expression levels. Actually, as it turns out, this particular gene is expressed at very low levels. And so we happen to have very deep RNA-seq sequenced libraries, and so we can detect it. But in other more publicly available databases, you don't always see expression of this particular transcript. And so if we looked at our lymphoblastoid cell lines that were genotyped for either homozygote major or minor alleles or the hets, we could see maybe some trend of a relationship between the SNP and RP1. It certainly does not meet statistical significance. There was absolutely no relationship between the SNP and IDLE, so this really didn't tell us much. So we wanted to know, okay, well, even though it's not an EQTL, maybe there is still some other evidence that this long non-coding RNA might be related to statin response. So we turned to our cellular system. And so here what we did is we decided to take tails from the LDL response distribution. In this case, we looked at the African American, high versus low, and the Caucasian, high versus low. Again, this is LDL response. We took cells from each of those individuals, treated them with simvastatin, and then performed RNA-seq. This was done by Debbie Nickerson's lab up at the University of Washington. So we had poly-A strand-specific libraries, and again, very deep sequencing, so 80 million reads per library. I think this is deeper than what most people do. And so if you look at the statin-induced change in expression of this non-coding transcript, we were quite surprised to see that in both the Caucasian and African American high responders, that statins actually increase expression of this long non-coding RNA. But if you look in the low responders, there's either a reduction or there's no change in expression. In contrast, if you look at MyLip or IDLE, you can see that statin treatment universally reduces expression levels of this gene, irrespective of phenotype. And so, this suggested to us, okay, we really should be looking at RP1. There's something special going on here. So one of the things that I didn't mention is that most of the genes involved in cholesterol metabolism are really well coordinately regulated at the level of gene transcription. And so oftentimes you can just see that transcript levels of a gene that you think might be regulated by cholesterol or you think might be involved in cholesterol are in fact sterile regulated themselves. And so if you take a human hepatoma cell line and then you sterile deplete by treating it with statins and a lipoprotein deficient serum, you can see that RP1 expression levels reduced. But then when you add back LDL cholesterol, we can see that that effect was exactly abolished. SRABP1 and 2 and LXR are some of the primary transcription factors which are known to regulate genes involved in lipid metabolism. And if you knock down SRABP2, we can see that there was a reduction in RP1. There appears to be no effect with knocking down SRBP1. And similarly, if we treat with an agonist for LXR, so this compound called GW3965, we can see that RP1 transcript levels are increased. And I showed here MyLip because MyLip is just a known LXR target. Okay, so what is the potential function of this? Given the fact that it was a pseudogene, there was absolutely nothing known in regards to if it had a function, what it might be. And so one of the things that we usually do is just ask, well, if we modulate its expression levels, can we see changes in terms of gene expression of genes that we think might be of interest? And so here I'm just showing you some genes involved in LDL uptake and some involved in cholesterol synthesis. And what we did is we transfected hepatoma cells with an overexpression construct containing either allele of that original GWAS SNP. And in the blue, you can see that RP1 overexpression increased expression levels of LDLR very specifically and robustly, but you can see that there's no effect on MyLip or idle, because one could imagine, just given the proximity of that pseudogene to MyLip or idle, that maybe it's regulating the gene that's close by. I mean, that's kind of a naive thought, but one might ask it anyway. One thing I wanted to point out here is that this upregulation of LDLR seems really specific. We don't see sort of a consistent increase in expression of all genes involved in cholesterol uptake or biosynthesis, and so therefore we think that it's not likely doing something to regulate the very well-known transcriptional pathways that just concordantly act across these genes. We can look at the functional outcome of upregulating LDLR, and that's looking at actual uptake of LDL into the cell. This is the fluorescently labeled LDL, and we can see that overexpression also increased uptake of LDL, and that the effect size is very similar in magnitude to what we're seeing at the level of the transcript. Because we know that LDLR actually is regulated at the level of transcript stability. And when you're looking at transcript levels, obviously it could be due to transcription or it could be due to decay of that transcript. We wanted to ask if there was any effect of RP1 on LDLR transcript stability. In fact, we saw no effect. And so this leads us to think that it is potentially possible that RP1 is acting to specifically promote transcription of LDLR through what mechanism we don't yet know. So I'm going to segue a little bit and tell you about a little diversion we had, and that was really to ask, is RP1 actually non-coding? So as we were making this overexpression construct, my text said, there's actually an open reading frame within this transcript. And I said, what? He said, yeah, there's this ATG site and there's a Kozak sequence, and even though it's only one exon, maybe it could be coding. And if you look at that GWAS, that SNP that was identified by GWAS, you would predict that this would make a protein that was 97 amino acids long. And the GWAS allele, actually, if this was protein coding, this would turn out to be a missense SNP. So what was originally thought to be a fully intronic SNP maybe was completely was annotated incorrectly. And so this is where I was like, well, wait a minute, I don't understand. How is something called non-coding versus coding? And so this is where I learned that a lot of the annotation algorithms that were used to define whether a transcript is coding or non-coding, because they're based on pattern recognition of various motifs, like the ones that Nadav talked about earlier, splice sites, initiation codons, poly-A signals, right? We understand that language. All of those various motifs are much richer in long open reading frames. And because of that, many of the programs decided we're going to err on the side of false negatives and call anything that was less than 100 codons long as non-coding. Now, that's clearly not a biologically defined threshold. That is an arbitrary threshold. And so we were really excited about this possibility. In fact, it actually had been reported last year in Cell by a group down at UT Southwestern that a muscle-specific long non-coding RNA actually functioned by encoding this very small, what they called a micropeptide, because this was maybe 33 amino acids long, that was fully within one of the exons of this long non-coding RNA. So we went back and we looked, okay, as I showed you before, if we overexpress RP1, we can see an increase in LDLR transcript levels. And we just did a very simple experiment in which we used cyclohexamide, which inhibits protein synthesis. And unfortunately, in the context of cyclohexamide, we can see that the effect remains exactly the same. And so this would suggest that protein synthesis is not required in order for RP1 to regulate LDLR. And so that might suggest that also RP1 itself does function as a non-coding transcript. But to me, this was a very educational experience. I can tell you that we have done Western blots looking for this. Negative Western blots aren't super exciting to show because you never know why they're negative. And I should point out that we do have a positive control. We know our cyclohexamide worked because we do see a reduction in total cellular protein. Okay, and so the model that we think is happening is that here I showed you that RP1 overexpression leads to increases in LDLR transcript, which increases LDL uptake and should reduce plasma cholesterol. In high responders, we see that statin treatment actually increases expression of this long non-coding RNA, and so that's consistent with the direction that we would expect. The high responders have greater reductions in plasma cholesterol. In low responders, we saw that, in fact, statins either reduced expression levels of this non-coding RNA or didn't impact levels at all, and so that's consistent with a smaller reduction in plasma cholesterol. We don't yet know how that original GWAS SNP that started us in on the story is actually related to RP1. I'm showing you a slide here that we see this time and time again that between the two constructs carrying either allele of the SNP, that the A allele looks like it has ever so slightly larger effect sizes than the G allele, but this never quite reaches statistical significance, and so we don't yet know if that GWAS SNP is related to RP1. And we also don't yet know precisely how RP1 is regulating LDLR. Okay, so the second story I'm going to tell you about today is, again, using the same model of trying to use the extremes in the response to identify a transcriptomic signature of drug response. And so we had reported a couple of years ago a very similar analysis in which we had used expression array data from LCLs that were derived from the Caucasian subset of the CAP population. And here we just asked, could we separate high versus low responders based upon endogenous gene expression traits? And so we used a method called non-negative matrix factorization non-negative matrix factorization to try to identify that separation. And so the output of that looks like this, where on the y-axis you're looking at a measure called purity, where 1 means you have perfect separation of the two groups. 0.5 would mean completely random separation. And the first thing we really wanted to do was understand, when you're looking at the tails of a distribution, what is the optimal experimental design? How many people, in terms of the N, should I include in my high responders versus my low responders? And so we titrated out the N. So here 20 means 10 high versus 10 low, because as you can imagine, if you have just small subsets of people, you can always identify differences between them, whether or not they're related to the phenotype you're interested in. So the red here shows our real high versus low responders. Our blue are just random responders. And so using this, we found that if we chose 26 high versus 26 low responders, we were able to maintain a very good level of separation. So this is 90% purity. So 90% of the time people were being attributed into the correct group, but with as minimal of a false positive rate as we could get. And so we were able to identify 100 signature genes, which you could see clearly distinguished these two response groups. But we wanted to extend this analysis a little farther. As I just told you, long non-coding RNAs represent the vast majority of the human transcriptome, and they're not fully captured with expression arrays. In addition, this analysis was limited to individuals of Caucasian ancestry, and we wanted to know, can we look at other ethnic groups as well? And lastly, to ask, if we look at variation in statin-induced change in gene expression, do we get a different sort of signal? And so for this, we used the same approach to try to identify a group of signature genes in first the Caucasian high versus low subset of the CAP cohort. And then we tested for cross-ethnicity validation within the African-American subset. And here we tried to sort of line up the phenotypes of high versus low to match. And so we identified 82 genes which best distinguished both the African American and Caucasian high versus low responders. And in looking at just a pathway analysis, we found that genes involved in glycolysis and gluconeogenesis and diabetes turned out to be enriched in some of these genes. And so this raises a whole other set of questions that I don't have time to talk about today, but which Karen touched upon, in that we think that there's some really interesting biology to look at the interrelationship between pathways involved in diabetes as well as cholesterol metabolism. So 13 of these 82 genes were actually non-protein coding genes, 7 were pseudogenes, and 6 were non-coding RNAs. One of the issues that we always have is, well, when you get this sort of result, how do you follow it up, right? I've got 82 genes. I need some other way of prioritizing what gene I want to study functionally. And so I mentioned to you at the very beginning that statins function by reducing intracellular cholesterol levels. So we thought, can we use cellular cholesterol as yet another trait to try to prioritize genes? So there's actually two types of cholesterol in the cell. There's free cholesterol, which mainly sits in membranes, and this type of cholesterol is really highly regulated and tends to not change. And so here you can see in either high or low responders that statin treatment has no effect on membrane cholesterol. In contrast, cholesterol ester is a storage form of cholesterol, and it's found within lipid droplets in the cell. And so now we can see that in our high responders, actually statin treatment is reducing the amount of stored cholesterol. And that's exactly what we'd expect to see. If you're inhibiting cholesterol synthesis, we should be reducing intracellular cholesterol. So we tested to see, could we correlate expression of any of those 82 signature genes with change in intracellular cholesterol. And we found that one gene called ZNF542P, the P standing for pseudogene, so again, another non-coding RNA, maybe non-coding RNA, was the most significantly correlated with the change in cholesterol ester. So here you can see that for cells for whom there was a reduction in ZNF expression levels, we had actually no change in intracellular cholesterol ester. And we could see that in our low responders, that statin treatment is actually reducing ZNF expression. And so this is all perfectly consistent with the fact that we saw no reduction in intracellular cholesterol ester in our low responders. So of course we know that correlation does not equal causation. When we knock down ZNF in a human hepatoma cell line, we can see not unexpectedly that there's no change in free cholesterol under either a statin or sham-treated condition. In contrast, if you look at cholesterol ester in that nice candy cane bar, you can see that when we knock down ZNF, we see that statin treatment actually increases intracellular cholesterol ester. And I would note that we do see a trend of an effect even under the sham condition, but clearly that this relationship is enhanced in the statin-treated condition, suggesting that statins are maybe increasing or decreasing some substrate that ZNF may be working on. And this relationship is perfectly consistent with the correlation that I talked about. So if you think about statin reducing ZNF as being sort of a very modest knockdown, so 0.8, this would be a 20% reduction. When we're knocking down with an siRNA, we're like at 0.1, right? So if this line increases, we would expect that knockdown should increase intracellular cholesterol ester. And so what I've shown you in this second little vignette is that we see that statin treatment reduces ZNF expression levels in our low responders but not high responders, that ZNF levels are correlated with changes in intracellular cholesterol ester, and that knockdown increases cholesterol ester upon statin treatment. And so what I think I've shown you today is that we have identified two long non-coding RNAs. Here again, my asterisks in terms of non-coding, because we haven't yet fully vetted whether or not ZNF may be coding or non-coding. Of course, there is an open reading frame. I think there is this bias towards studying poorly annotated genes, and we know that the vast majority of long non-coding RNAs really are poorly annotated. We don't know that they have any functional effect at all, and so people don't really want to look at them. It's a lot harder, and so I think that there is really substantial work that remains to evaluate the role of long non-coding RNAs in pharmacogenomics. And so I very, very briefly just wanted to touch on what I think are some of the challenges in evaluating the role of long non-coding RNAs in pharmacogenetics. And I think the first is this issue of whether or not it's coding or non-coding. And I think one of the biggest issues here is that the peptides, which might be generated by these small open reading frames, oftentimes these are missed by proteomic screens because those screens are really limited to looking for larger peptides. If you do have a peptide that's generated by one of these short open reading frames, they tend to be not very abundant in the cell, they are rapidly degraded, and ironically, the functional outcome may or may not be due to that translated product. Just the act of that open reading frame sitting on the ribosome, that might be the function of the long non-coding RNA. And so the function of the long non-coding RNAs, we think about microRNAs and they all pretty much do the same thing, but you cannot have that same assumption for the long non-codings. They don't behave the same way. Anything you can imagine that they might do, there is some example of it doing. Loss of function studies are really hard because of the way that these can be contained within the genome. If you have something that is generated antisense to another transcript, you can imagine if you knock out what you think is your long non-coding RNA, you could be knocking out lots of other genomic features. And you can't use those simple tools like just inserting a premature stop codon. And so at this point, we're really limited to antisense oligos. And so lastly, I just wanted to very briefly mention that we have this issue of conservation, that a lot of the long non-codings are not well conserved in non-primate mammals. In fact, maybe 20% of them could be human specific. And so when we think about that gold standard of translating to physiological relevance, that's not going to be straightforward with a lot of these guys. And so I want to sort of leave with this question of thinking about when you stumble upon your long non-coding RNA in your research, and I think you will, because we know that these guys are the vast majority of what's transcribed, how are you going to follow it up? Are you going to use things like conservation to try to prioritize? Because some people argue lack of conservation is lack of function, but lack of conservation could just equate to some sort of adaptive function that is specific to humans, and it could still be very much important. And so with that, I would like to acknowledge a number of individuals worked on this project. Ron Krause, of course, for spearheading all this work in pharmacogenomics for a number of years. Beth Toish, Kungpil Kim, and Katrina Mitchell were the three postdocs that worked on the RP1 and ZNF projects. And I'll take any questions. Thank you.
Hi. So with regards to your first statin-associated link RNA, so you were looking for this potential EQTL effect. So I was wondering if you've looked at any of the new GTEx EQTL data on all the different tissue types, just because you never know what cell type it might be coming from. And I think you said that the sentinel SNP is exonic, right?
Okay, so if that SNP is within that transcript.
Right.
Yes.
So I'm wondering if you maybe, to sort of get around the issue of not being highly powered for a really, really small effect size, if you had any sequencing reads in individuals that were heterozygous for that SNP, and if you had enough depth to see if allelic imbalance within an actual heterozygous individual.
Absolutely. So that was something that we did try to do. Unfortunately, our sequencing reads, even at that 80 million reads per library, were not deep enough to look at that because I don't think I emphasized how rare this transcript actually was within the libraries that we had. We have gone back and looked at GTEx. We haven't seen any evidence that that SNP is an EQTL for either of those two genes.
And just the last thing I would say, just because I did a lot of this for my own project because of this issue, is that you can now obviously use these, exome capture probe pools to pull down your exons of interest, but I'm not sure if they'd have it for a transcript like this. So.
Right. I think, again, a lot of the tools that are available are biased towards protein coding genes and things that people think about as being regular ORFs.
So kind of following on that, I was really excited about your RP1 story, and also really disappointed that you guys concluded that it wasn't a coding. So,
as was I.
So you used the cyclohexamide to convince yourself that it was the case, but as we know, it inhibits the synthesis of protein by inhibiting the activity of ribosomes. So there are good chance that the proteins are there. I mean, you showed that the cyclohexamide was working by looking at the whole protein level, but so is there any follow-up experiment that you guys are thinking about? Maybe taking the sequence to the expression cassette and maybe introducing synthetic amino acid that has the same sequence and see if it has more effect?
Yeah, no, absolutely. So the one caveat I should have said is if it turns out that those proteins have long half-lives, if they're very stable, the cyclohexamide obviously will not have any impact on that. And so, yes, we could absolutely do that simple experiment of just trying to introduce a premature stop codon or disrupting the ATG. When you see sort of negative data, right, you become less excited to take those next steps.
Okay. All right.
But yeah.
That's fair.
Thank you.